what is episodic memory and how do we study it comes in many forms. I'm just giving you one variant on this that has to do with a core computation in episodic memory that I would argue is really necessary for you to be able to keep track of experiences. And that stems from the notion that memories that we experience on a daily basis are highly overlapping in their nature. So think about parking your car in the parking lot, for example. Every day you park your car in the same lot. You might park in the same vicinity, maybe close to an entrance. Yet every day when you go out to the lot, you remember where you parked your car today. Hopefully you're not going to the same spot where you parked it yesterday. These memories are highly overlapping. They have so many similarities with each other, and yet the brain does not overwrite these memories on top of one another, has a way to be able to keep them separated from each other. So how does that happen? And the answer is a neurocomputational process that is called pattern separation. And the argument here is that this is a necessary episodic memory process by which we can distinguish among similar memories, and I'll talk a little bit about the mechanisms, but it really appears that it requires the hippocampus, at least in its most exquisite form, where you're distinguishing among what, where, and when. Those conjunctions seems to require the hippocampus. So how does this happen in the hippocampus, or at least how do we think it happens in the hippocampus? Now, answers to that come from the work and modeling by David Marr, who's pictured here on the left next to Tommy Pojo. There's very few pictures of David Marr out there, but I like to always point out that he unfortunately died at a very young age, but not before he could make incredibly seminal contributions to the field of neuroscience and cognitive science. And what he hypothesized, or what he thought, was that within the hippocampus, we've got two subfields in neighboring vicinity, the dentate gyrus and the CA3 region, that have architectures that are really exquisitely suited to carry out pattern separation, and the flip side of it, which is pattern completion. So on the top left here, you see a simplified schematic of hippocampal wiring. You see that input comes in from the entorhinal cortex through the perforant path, gets to the dentate gyrus as well as to the CA3. The dentate gyrus projects to the CA3 through the mossy fiber connection, and then CA3 projects to CA1. CA1 gets additional input from the entorhinal cortex, that is the monosynaptic route that is separate, and then output funnels from CA1 and subiculum back out to the deep layers of the entorhinal cortex. So that is sort of the hippocampal side loop of processing. Input comes in from the entorhinal cortex and funnels back out. So how does this accomplish pattern separation? Well, it turns out in the dentate gyrus, you have a really interesting and unique architectural feature that has to do with the density and type of cell that's there. So in the rat, for example, there's about 1 million granule cells that are very small, densely packed cells in a dentate gyrus. And that allows the 250,000 or so pyramidal cells in the entorhinal cortex to project a signal out and allow that signal to essentially be pattern separated, represented across a sparse pattern of firing cells. And we know this to be true about the dentate gyrus. It's very difficult to get cells in the dentate gyrus to fire, so it has a sparse coding scheme, which supports this process of pattern separation. The CA3 is an interesting battleground, though. It gets input both from the dentate gyrus as well as from its recurrent collateral connection or recurrent collateral network. So this is a massive autoassociative or potentially autoassociative network in the CA3, and provides a chief source of input to the CA3 region. Now you can imagine you can get a lot of runaway excitation here with this recurrent collateral network. So there's a lot of active inhibition in this region also. But the idea is that if the dentate gyrus has a separation signal in it, it can project that signal through the very powerful mossy fiber connection onto the CA3, where the CA3 could also have its bias reflecting pattern separation. But in the absence of that signal from the dentate gyrus, the CA3 is left to process using its own recurrent collaterals and reflects much more of a recognition signal or what's called pattern completion, the ability to fill in or retrieve an experience based on a partial or a degraded cue. So here you have a structure in the brain where the the architectural features that are represented here can give rise to these computations. But it wasn't until many, many years later, in the early 2000s, that there's been an accumulation of data suggesting that, in fact, this is what's happening in these networks. And these initially started with single unit recordings in the CA3 and CA1 regions, and then eventually the dentate gyrus. And then were followed up on by functional MRI work that also showed that in humans, these subfields, dentate and CA3, seem to be involved in this pattern separation computation. And now we have a fairly good understanding that these subfields are really essential to carry out that computation. So there are patient cases, for example, where the dentate gyrus is missing or is lesioned, for lack of a better term, and they do struggle on tasks that emphasize pattern separation. I'll show you one example of those tasks momentarily. So one of the things that is always an interesting approach to study a phenomenon is to ask about situations in which it breaks down. And I mentioned to you before that my other passion, in addition to memory, is trying to understand the aging brain. So I had a really unique opportunity here in the work of Michaela Gallagher and Howard Eichenbaum and Heike Tanila and some other collaborators in the early 2000s, where they had showed that this pattern separation computation is potentially quite impaired in older rats. So these are rats that were exposed to environments, as you see here. Lots of similarities across those environments, but the geometry is different. And if they were recording from the CA3 cells in young animals, what they found was that these young CA3 cells could remap readily. So they were able to distinguish among those similar but not identical environments. But if recording from an aged CA3 place cell, there's a failure to remap. And here you can see there's a lot of overlap in the representation across those two environments, suggesting that this cell may be oblivious to the fact that these environments are different. In other words, it's a failure to pattern separate, or an overgeneralization or pattern completion process at the expense of pattern separation. So this became an inspiration to try to ask the question, well, do we see any of this happening in humans? And of course, we had some circumstantial evidence in humans, because we know that in aging, there is, of course, a memory loss. Older adults struggle with episodic memory, with source memory, with recollection, with contextual memory, spatial memory. There's even an increase in false memories as we get older. And we argue that actually all of these things could have one element in common, is that they could be driven by an impairment in pattern separation. But circumstantial evidence, as I'm sure you will all agree, is not sufficient. You need to test this experimentally. So we designed a task, and this was work that I did with Craig Stark back in 2008, 2009. We started to develop these tasks that taxed pattern separation. And the way that we did this was using stimuli that were highly similar to each other, but not identical. So one example here is the seahorse. It's a terrible joke about the hippocampus and the picnic baskets. So if you look at the picnic baskets during the study phase versus the test phase, you'll see that they're similar but not identical. And of course, if the test phase comes, a few minutes or 20 minutes or so after the study phase, and I ask you, have you seen this exact picnic basket before, or did you see something similar to it? That's a very challenging question. So challenging, in fact, that even young adults didn't do as well with it. So here you see the data that we collected in young adults and older adults, so 20-year-olds and roughly 70-year-olds. The first feature, you'll notice that if you look at brand new items, novel items, or repetitions, exact repetitions like the four-leaf clover, there's virtually no difference among the populations in terms of the response distributions. But if you hone in on the lures, the similar items, the ones that are similar but not identical, like the picnic baskets, you see that even the young adults struggled with them. They generated a lot of old responses. About 30% of the time, they falsely recognized that item and said, oh yeah, that's the same picnic basket I saw before. But importantly, the older adults made that mistake much more often, and they were more impaired on this task, but only when you select for the lures. And this is a really, really important thing to keep in mind, because if we didn't have the lure stimuli and just had novel versus familiar recognition, the conclusion from this experiment would have been that there's no age-related memory loss. But it was the lures and the way that they challenged the hippocampus and this pattern separation computation that allowed us to see this overgeneralization bias in older adults. Importantly also, we identified a neural basis for this. So it wasn't sufficient to just look at it behaviorally. We needed to understand the mechanism that might have led to that. And we knew that the putative mechanism from the animal work that I mentioned earlier was a hyperexcitation in this recurrent collateral network in the CA3 region that resulted in higher levels of activity and a potential bias towards pattern completion. We found the same exact thing with high resolution functional MRI while participants were doing this task. And we also determined that the level of activation was related to their performance on the task. In other words, the more hyperactive this region of the hippocampus was, the worse off their behavior was. And this was really important because it was a counterintuitive discovery with respect to what else has been shown in the human literature. So we actually struggled quite a bit to get this published because no one really believed it. No one believed that higher levels of activity were an index of dysfunction rather than the more commonly adopted account at the time, which is compensation. It's just older adults ramping up their neural resources to be able to solve the task. But the beauty of this is that, you know, you do something in your lab and then you hope that others replicate it, and this has been replicated now by 7 or 8 different labs around the world. So we are fairly certain that hyperactivity is pathological. But even more so, if we do believe it's pathological, the question is what happens if you try to reverse it? And that's exactly what we did. This was, again, collaborative work with Michaela Gallagher and Arnold Bacher and Craig Stark and some other collaborators, where we used that same task in the scanner, but in this case, we looked at healthy older adults and patients with amnestic mild cognitive impairments. One of the first things that was identified is that they also expressed this hyperactivity in the CA3 indentate. You see that in the first 2 columns, the first 2 bars, the white and the black bar. Those are AMCI patients on placebo. But those same patients were also given, in a different phase of the trial, a low dose of an anti-epileptic. Now, these are not epilepsy patients, remember. These are just amnestic MCI patients, but we believe that they have this paradoxical and pathological, potentially, hyperactivity, and we're trying to reduce it. So that's exactly what happens, and the level of activity was normalized. But importantly, also, the number of false alarms or false recognition trials also went down. So not only did the level of activation go down, but also performance or behavior was enhanced. And this was very, very exciting. This was a phase 2A proof of concept clinical trial. And now my colleagues, Dr. Gallagher and Dr. Bacher, are in the throes of a phase 3 clinical trial that I hope is not held up too much by COVID-19, to be able to test this fully in AMCI and may lead to a brand new type of treatment or therapy for patients in the earliest stage of Alzheimer's disease. So this is the story in terms of functional activation, but then we asked, well, what about structural degradation? Could this be part of the story as well? And we knew that some of the other elements of the hippocampus network are compromised with age. Namely, there's been a lot of data in both in pathological cases, so looking at histopathological evidence from AMCI patients and Alzheimer's patients, as well as in animal models showing that the perforant path that takes information from the entorhinal cortex and provides it to the hippocampus is compromised in the aging process. So we hypothesized that we might be able to see this actually in older adults using non-invasive imaging techniques. And that's exactly what we did with diffusion imaging. So Craig Stark and I developed an approach that we called ultra-high resolution diffusion imaging. And we used that to look at the integrity of the perforant path. I'm not going to go through too much of the detail on this, but we showed also a little bit later that the kind of tractography, probabilistic tractography that we were getting from our 40-minute sequence, that's what you see in panels B, was commensurate with the kind of connectivity or tractography that our colleagues up at MGH, Gino Gustinac and others, have been able to do with postmortem tissue on a much stronger magnet for about 40 hours of scanning. So here we had a clinical grade sequence that we're able to measure this biomarker. And indeed, we were able to see that the perforant path signal was degraded in older adults. And I'm not showing it here, but the degree of impairment was also correlated with their performance deficits on this pattern separation task. So we started to put the pieces of the puzzle together. And this is based on some modeling work that we put in a review. My former student, Stephanie Leal, who's now an assistant professor at Rice University, and I put this paper together for Trends in Neuroscience in 2015, where we talked about the core vulnerabilities in the aging medial temporal lobe. And the idea here is that it starts with a loss of inhibitory tone in the CA3 network, and in part also in the dentate network. And as a result of that, you get CA3 hyperactivity, which is this emphasis on the autoassociative fibers, this recurrent collateral network in the CA3 region. That may cause some dendritic retraction, these apical dendrites from the CA3 that link up with the perforant path. And as a result, you might start to see perforant path loss that diminishes the quality of the information coming in from the entorhinal cortex and to the dentate and CA3. So what this leads to is essentially an isolation syndrome, where the hippocampus is now isolated from the input that is coming from the rest of the brain, and as a result, it is unable to process memories efficiently. But we needed to also assess this part of the network. We needed to understand what is happening upstream of the hippocampus and whether that is also compromised. So in other words, is it truly something about the hippocampus, or is it mostly just garbage in, garbage out? In other words, the quality of what's coming into the hippocampus is already compromised, so we can't possibly expect the hippocampus to do a good job. So this was the next assessment that we embarked on. And some of you may know this already, but the entorhinal and perirhinal impaired hippocampal cortices are not one size fits all. There's a division of labor there. So there is a domain selectivity that follows, roughly speaking, the what and where pathway. So perirhinal cortex and lateral entorhinal cortex seem to preferentially be responsive to object properties, whereas perihippocampal cortex, or postrhinal in the rat, and the medial entorhinal cortex are preferentially responsive to spatial information. So we needed to design a manipulation that allowed us to parse this division of labor and try to understand it. So now we shift from the translational, going back to fundamental, and trying to understand these computations. And this was the work of Zach Ray when he was a student in my lab. He's now an assistant professor at WashU St. Louis. So my students are starting their laboratories and starting to do some really excellent work. But this was the work that led up to his dissertation. And he designed a task that pit against one another the notion of pattern separation based on object identity versus pattern separation based on spatial location. It's a very simple manipulation where participants would see an object on the screen in one of many different locations, and during tests, and they're just simply asked to respond with, is it an indoor item, and do you see it on the left or right? Indoor, outdoor, left or right? And then during test, they either saw the same exact item in the same location, so that's a repetition. They saw the item change identity, but not spatial location, so that would be an object lure, like the chair. Or the same item has not changed identity, but has moved. So that's a spatial lure, changes spatial location, or a brand new novel foil that they've never seen before. And you can imagine this task would be very, very difficult, but in fact, Zach worked very hard to make sure that conditions are equated in terms of difficulty and was able to use this to test young adults and older adults. We'll talk first about the young adults. First, we needed to establish that there's a division of labor in the medial temporal lobes with respect to this task. So he looked first at the object lures, the object correct rejection, and hypothesized that the modulations that we would see in this region would involve the lateral entorhinal cortex and the perirhinal cortex, the putative object network, and of course the dentate and CA3 in the hippocampus, as we'd seen before. And that's exactly what he found. And then the question is, if we now switch over to the spatial lures, the spatial items, do we see the flip side of this? Do we see engagement in the putative spatial network? And that is exactly what he saw. When he looked at the spatial lure items, it was really the medial entorhinal cortex and the perihippocampal cortex that showed these modulations, and not so much the lateral entorhinal and perirhinal cortices. Again, the hippocampus was engaged. Turns out pretty much any sort of separation tends to engage the hippocampus. I won't go through too much more data from that paper, but I just want to show you the model that we proposed at the time, which was not entirely novel. There was a lot of evidence suggesting that this would be the case from Tim Bussey and Lisa Saxida and others, showing that there's a hierarchical processing of interference in the medial temporal lobes. And here the notion is that the pattern separation form that happens in the medial temporal cortex might be domain selective. So it may be very much aligned with the what and where pathway. But when you get to the hippocampus, that's where you get the colored segmented bar towards the bottom. That's when you get the most pristine form of this pattern separation, full disentanglement of the experiences, and it is domain agnostic. So it really doesn't matter if it's what or where or any other parameter. It is really experiential at that point rather than locked to a particular dimension or parameter. So that was the foundational understanding that we tried to get to. And now the question is, well, what happens when you start to look at older adults? So our logic and rationale here was based on a lot of prior data. Again, we're very much informed by cross-species evidence in this case. And we knew from work by Sarah Burke and Carol Barnes that object recognition deficits manifest in PRC perirhinal lesioned animals. And there's an abundance of this in aged rodents. We knew that layer 2 of the entorhinal cortex, in particular the lateral entorhinal cortex, was a site of a lot of pathological changes, including a loss of reelin and a buildup of phospho-tau, which is a precursor to building tau tangle pathology in Alzheimer's. And this was work from Michaela Gallagher and other colleagues. We also knew from Brock and Brock that the lateral entorhinal cortex and the perirhinal cortex are among the initial sites of tangle pathology in Alzheimer's disease, and that's what's called the transentorhinal region. And then finally, more recent data from Morgan Behrens and Rosanna Olson's labs showing that the anterolateral entorhinal cortex structural integrity, so volume, is correlated with cognitive decline in undiagnosed older adults. And I'll show you some data also from us that showed that in MCI patients, we see something very similar. In fact, it turns out to be a very, very well-behaved biomarker that helps pull apart those with more impairments. So given these data, given these kind of suspicions that the lateral entorhinal cortex might be the place to look, Zach embarked on a testing exercise in older adults, again, comparing young versus old, and using the object and the spatial memory tasks. Here, separated them out. So we had blocks of object and blocks of spatial. And what he found was evidence for hypoactivation in the anterolateral entorhinal cortex that was not there for the posterior medial, and was only there also for the object pattern separation trials and not so much for the spatial pattern separation trials, suggesting that there's domain specificity and there's regional specificity. So really, it's the anterolateral entorhinal cortex that seems to be compromised. Now, going back to our core vulnerabilities model, putting the pieces back together in this puzzle, we have C3 hyperactivity, we have perforant path loss. Now we can add on another condition that is lateral entorhinal cortical dysfunction. And the logic here is that this could be of translational value, because if we can figure out a way to, for example, reduce the hyperactivity, say with a low dose of an anti-epileptic or any other number of potential methods, we could potentially enhance either the quality of the inhibitory tone or the ability of the CA3 to listen to that inhibitory tone, because it won't be washed over by some of this hyperexcitability. And if we're able to do this successfully very early, it may be the case that we are able to rescue or prevent some of the damage to the perforant path, and perhaps kind of by a backwards route, be able to rescue LEC dysfunction. I'm not showing you these data here, but if you go back to the clinical work in 2012, that Neuron paper, Bakker et al., you'll see that one of the other things that the anti-epileptic was able to do was increase the level of activity in the entorhinal cortex. Now, it's difficult to tell whether that is because of what happened downstream or what happened upstream, but right now we have this really interesting rebalancing of the network that happens with the reversal of hyperactivity. So I know we talked a lot about the aging brain, and I showed you a way that this can be translated and applied to some of the earliest stages of Alzheimer's disease. But we want to really be able to look at Alzheimer's disease directly to see if we can find evidence of loss in these regions. So this brings me to the work by Andrew Holbrook and Dan Gillen, and a paper that we published just a couple of months ago in Alzheimer's and Dementia. This is based on the ADNI study, the Alzheimer's Disease Neuroimaging Initiative, very large data set. We had developed segmentations for the anterolateral entorhinal cortex and the posterior medial entorhinal cortex, and asked a very simple question about whether cortical thinning in these regions is going to be predictive of trajectories of decline in control participants, MCI participants, or Alzheimer's disease participants. And on the left here, you see that the ALEC was much more discriminative of these groups and was able to show that decline in a much more delineated way. Andrew also did a really interesting analysis on a subset of those individuals who also had CSF lumbar puncture, so we could evaluate their pathological status, both A-beta and phospho-tau, calculated something called the amyloid ratio measure, which pits those two against each other. It's a well-known measure of Alzheimer's pathology, and what he observed was that the anterolateral entorhinal cortex thickness was well differentiated by amyloid ratio, whereas posterior medial was not. So all of this was to say that the anterolateral entorhinal cortex thinning could serve as a really nice biomarker to track both cognitive status and potentially longitudinal decline over that 30-month period. He then went on also to look at how it's associated with memory scores. So we had the clinical dementia rating, the memory subset in particular, as well as the mini mental state exam. And in all cases here, it was really, I'm going to call it ALEC and PMEC. So ALEC was much more able to predict change in these scores, and change in ALEC was much more able to predict these scores versus PMEC in this case. And then another analysis is just using ROC curves. So this is a receiver operating characteristic to try to predict MCI status in AD using nothing except for ALEC and PMEC cortical thickness. And again, you see ALEC is outperforming. So this all told us very clearly that looking at the anterolateral entorhinal cortex could be a structural biomarker that is an indicator of decline, potentially because of cell loss in that region, potentially because of the aggregation of tau pathology, which we already know happens predominantly in that region in the early stages. So now, based on this, one can go back to our understanding of the what and where and so on, and start to ask questions about, well, what about the other side, the spatial side, right? This is all very focused on the object network. What about this spatial network? And this is the work of Freddie Marquez in the lab, which hopefully that paper will be out very soon, so you'll see more details on these experiments. I'm just highlighting a couple of data points here. Using the spatial task, which, as I said before, is really a task of discriminating across spatial location, and testing in a sample of older adults with a lot more heterogeneity in their memory performance. So it includes some amnestic MCI patients. What he found was, if you looked at the resting state connectivity in the posterior medial network, he could identify essentially a network-related mechanism for performance of this task. This is what you see here. PM is posterior medial. So posterior medial functional connectivity averaged across all of the network nodes was positively associated with the discrimination index in the spatial task. So this is a circuit-specific task performance. And then he went on to show two really interesting associations. One of them is with total tau. This is based on CSF, where you see that there's an inverse relationship between total tau and CSF and spatial blur discrimination, so performance on the task. And a positive association with A-beta 4240. Keep in mind that because this is in CSF, the higher that ratio is in CSF, the lower it is in the brain, and vice versa. So here you see a positive association with PM functional connectivity, suggesting that the lower or the higher the functional connectivity, the lower the brain's CS or the brain's amyloid level. So in this case, there was a differentiation in terms of the types of pathology that are being reflected either in a circuit biomarker or in behavior. And the upshot, and without going into too much more detail, is that the task performance was associated with tau, which we've typically seen when we've looked at tau in the past. Tau seems to be much more directly linked to cognition than amyloid is. But very interestingly, amyloid level was correlated with or associated with functional connectivity in this posterior medial network, and that was not connected with tau. So you can start to kind of infer what the bigger picture here might be, that there may be some functional changes in this network that are related to amyloid aggregation, which we know manifests in the posterior medial network. And then perhaps the manifestation in behavior is really driven by the tau aggregation, which is considered a marker for cell loss. So we're still kind of putting all the bits and pieces together for this story, but it's an exciting one, and it suggests that this task is also sensitive to some of the early stages of Alzheimer's disease, albeit maybe a little bit later in the game when you start to see real cell loss rather than just functional decline. Okay, so to summarize the pattern separation results in aging so far, pattern separation is a critical episodic memory process that requires the hippocampal dentate gyrus and CA3. Object and spatial pattern separation engage parallel pathways in the medial temporal lobes. We've talked about the what and where pathways. The anterolateral entorhinal cortex appears to be selectively vulnerable to aging and is associated with object discrimination deficits. And then finally, the posterior medial network, which is involved in the spatial discrimination, seems to be associated much more overtly with Alzheimer's pathology. So this is kind of where you make the leap from aging to Alzheimer's. So this notion that we could really study circuits in the brain and understand both pathology and conditions of deficit or dysfunction, as well as conditions of improvement, like treatment, for example, using these pattern separation tasks and neurobiological investigations that are surrounding them, became a real big theme in my lab. And just to kind of give you a little bit of a rundown, I'm not going to go through all of these results, but they may be things that pique your curiosity. But in the early 2010, 2011, we were still putting the pieces together for the aging and Alzheimer's model. I've shared a lot of these data today. Later on, we tested the treatment model, and we saw that that can improve pattern separation, and now that's in phase 3 clinical trials. A little bit later on, we also looked at the impact of caffeine. For example, a mild dose of caffeine was able to enhance performance on this pattern separation task when individuals received the test 24 hours later. This was a post-learning administration. A couple of years later, Stephanie Leal and I did some work together on identifying neurobiological basis for depression that really brought in also the contributions from the amygdala, not just the hippocampus. And she developed a beautiful emotional pattern separation task, which I didn't really go through in detail today, but was very informative with respect to what's going on with depression and the idea that there's a negativity bias that might involve actually a hyper discrimination of negative information. And just a couple of years ago, we also discovered that very mild exercise, so on par with walking, and very short dose of that, like 10 minutes, can also improve pattern separation in healthy adults. And now we are seeing evidence that this is also the case with a longitudinal intervention in older adults, and that data hopefully will be out in press soon. So very, very productive set of approaches, and very diverse and versatile in terms of their applications. And certainly many of our colleagues have used these tasks to look at sleep, to look at schizophrenia, to look at a variety of different other conditions. And we're very happy to see the widespread adoption. Now the question is, what about the future? What is it that the lab is really focused on now? And I talked about it a little bit, and Shantanu brought it up in terms of biomarkers, but I just want to give you a glimpse of what our future is looking like right now. And it's inspired, of course, by this global challenge that brain disorders are devastating, and they affect a very large proportion of the world's population. And yet, given this, and given the number of resources that we're throwing at this problem, 85% of clinical trials or drug trials in CNS in brain disorders have failed. And there are a lot of reasons why these failures might happen. I'll give you some examples. Maybe there's an incomplete understanding of disease circuitry. Maybe there's poor translation from preclinical models. Maybe there's a focus on pathology rather than circuits, and possibly pathology like amyloid may not be the best target. That's an arguable point, of course. Maybe there's uninformed selection and stratification because we lump patients together under the same diagnostic umbrella where in fact there's a lot of heterogeneity. The links between disease features like proteinopathies and so on and cognition are quite poor in some cases. And outcome measures in clinical trials can also be quite insensitive. If we're talking about a global cognitive or clinical measure, typically that's not going to be sensitive to minor deflections. And finally, there's a lot of investment in clinical trials, of course, but there's insufficient de-risking of clinical trials. And that de-risking can happen with neurobiological validation, but there's not enough emphasis that is focused on this. And as a result, you see lots of things that go on to phase 3 and unfortunately fail after investing $100 to $200 million. So how can we address this? What are we trying to do? And this is what I'm calling the Biomarkers plus Cognition platform, or the BC platform. Now, the idea here is that we couple the sensitive cognitive tests that we have been developing, which allow us to really detect very subtle changes in cognition, with circuit level biomarkers. These could be functional, they could be structural, connectivity, diffusion. We kind of throw a whole boat of different approaches at it and select which biomarkers are going to be best based on the indication. But as a result, if we're able to do this validation very early, we can substantially reduce risk in pivotal trials and increase the probability of clinical trial success. And I'm not going to go through this in detail at all. I know it's a busy slide. I'm just kind of showing you that we've developed now a number of these tasks. I've talked a little bit today about the object and spatial tasks. But we also have a temporal discrimination task and an emotion discrimination task. And there are several others that we're working on now, including reward processing and other facets of memory. All of these can be fodder, can be input into a model that allows us to improve diagnostic accuracy. And in terms of diagnoses, there's a lot of different markers that can be used. Some of them are molecular, and molecular, as you know, struggles with a lot of problems. They could be nonspecific, invasive, not always linked to cognition. Certainly, we've seen that. Or clinical assessments, but that could be too crude. It could be too late by the time patient has symptoms. It may be much later in the game than we'd like for us to intervene. Also based on humans, so of course it's error prone and it's expensive to have a full clinical workup. Cognitive markers could be sensitive, could be specific, could be reliable, and could be done cheaply, and especially in digital format. And circuit biomarkers could also be sensitive and specific. They're non-invasive for the most part, and they're linked to cognition. So our focus is really on the combination of cognitive and circuit markers, and not so much on the molecular and the clinical. So we're trying to develop an approach to take these together and use artificial intelligence or machine learning to do what is termed digital precision diagnostics. So here you kind of get a glimpse of what this is like. We have a number of tasks that are performed. We have a lot of feature extraction that we use to various different methods to be able to extract useful information out of performance on these tasks. And of course, there's neurobiological validation that went into these tasks, so we can attribute it to particular circuits. And now we're using a convolutional neural net approach. This is work by John Jancic in the lab to be able to output diagnosis. And of course, you need a lot of data for this, but this is what we're working on. So how do we do this? Well, the first stage is validation. Like I said, we've done a lot of work in terms of biological validation with circuit-based imaging in small, well-characterized samples. So this, we've accomplished quite a bit of that, at least in the primary indications of interest. And now we're in the process of collecting massive data sets. So now that we have a platform, the idea is to test and monitor using these tasks on digital platforms in a ridiculously large number of individuals with a lot of heterogeneity. The idea is that heterogeneity is going to be really good for us because it will allow us to train our models and allow it to diagnose very subtle impairments based on the combination of cognitive and biomarker profiles. So that's sort of where we are now. We're trying to build out these large data sets and see if we can benefit from the tremendous advances in AI to come up with better precision diagnostics. And again, if it weren't for the foundational knowledge and the circuit and neurobiological understanding of learning and memory, of course, none of this would be possible. So I hope that the major take home here is that understanding the fundamental inner workings of a brain system allows you to then translate it in a very informed way. So with that, I just want to thank all those who have contributed to the work. I've highlighted certain individuals who have been key to some of the data that I showed, but of course, the lab is a big family and everybody contributes to everything. And here you see one of our most recent lab pictures, unfortunately, is a Zoom picture because we couldn't get together in person. But, they're the ones that really do all the work, and I'm just able to maybe talk about a little bit in advertising. So with that, I want to thank you for your time, and I hope that we have a little bit of time for questions. Thank you.
Michael, excellent talk, very inspiring. I have a few questions on my own, but I can wait for the audience to ask. And if you want to unmute yourself and ask the question, just do it. Or you can type it in chat. So, Mike, can you see the chat?
I see one in chat, right? One from Colin. Could you speculate on whether CA3 autorecurrent hyperactivity could play a role in the phenomenon of deja vu? Interestingly, yes. So at the end of the day, I don't know exactly what the mechanism for deja vu is. I think it's very difficult to say whether there's one particular mechanism for it. But that is one hypothesis, is that it is this very strange, compelling sense of extreme familiarity with the experience that results from a faulty pattern completion. So in other words, if a partial or degraded cue gets recognized as exactly the same experience that happened before, depending on how powerful that attractor is, you could potentially have a feeling of deja vu. What is more difficult to explain is the emotional feeling, the compelling feeling that comes with that extreme familiarity, that you could almost swear up and down that you've seen this before. So that I don't really have a good explanation for, or the whole thing could just be a glitch in the matrix. I'm not really sure. Somebody just raised their hand. Ian?
Yeah, hey. So cool stuff. I had two questions. For the first one, for the distinction of like the what and where pathways, I guess I had always like learned of them and thought of them as the more naturalistic position in an environment, sort of kind of 3D space. And then the cues that you're using are very, I mean, kind of like non-naturalistic in that they're on a screen in a different spot. So could you talk more about that and how that's still okay and whatnot?
Yeah, no, thank you. So it's a great observation and a great question. And of course, what you'll notice in most of our designs is that we have simplified to the point of stupidity, right? I mean, it is stupid simple. And there's a reason for that, Ian, right? Because when we take something like our object manipulation or our spatial manipulation, we can do the same exact experiment in a rodent. That was the inspiration. I'm not saying that, rodent 3D is not possible. Of course it is, but it's much more difficult to really try to understand that. So when we first designed these experiments, they were designed around the notion that if we reduce to the point of almost absurdity and try to find the root computation here, we might be able to find elements of neural activity that are well differentiated. And that's exactly what we found. I can tell you about some of our more naturalistic work that involves the other parameter that we didn't talk about. We talked about what and where. We didn't talk about when. So work from Maria Monchal in my lab just a couple of years ago showed that you can do a very naturalistic movie viewing kind of task. And based on that, you can also determine that there's a functional division of labor in the medial temporal lobes. Turns out the anterolateral internal cortex also plays a big role in time and time processing. But it was so much more difficult for us to parse that than it was for the object and spatial, right? That beautiful distinction that you saw, that almost like a change detection exercise across the networks, that I would argue happened because of the simplicity of our design. Now, do I believe that from that we can go straight to real life experience? Probably not. We still need several steps along the way. And we're doing a lot more naturalistic work now in the lab, but we needed that fundamental basis because that's how we can bridge with the animal data. Does that make sense?
Yeah, that makes a lot of sense. Thank you.
Of course.
The other thing I had a question about is the discussion of biomarkers in the clinical studies. So would the idea be to limit recruitment based on biomarkers or base outcomes of the trials on biomarkers?
Yeah. All of the above. So there's a few different ways that it can be helpful. One is the idea of stratification, right? You want to be able to select and stratify different patients based on some evidence that tells you that they have an ongoing disease process. Right now, for example, if you're doing a study, a clinical trial in preclinical Alzheimer's disease, so those who don't have manifest symptoms yet, but maybe have risk, typically that's based on some biomarker evidence, like they have amyloid in their brain. But we know also that 30% of older adults with amyloid in their brain will never get Alzheimer's disease. So it doesn't have that specificity. If we had a circuit biomarker that was much more specific, that could tell us in the preclinical or asymptomatic stage more definitively that certain patients are going to go on the path to Alzheimer's and certain patients won't, now we can really constrain how we do this study. One, it saves a lot of resources, and two, it actually works out better in terms of being able to establish the effect, because if you know that everybody's going to get it, you have a much better handle on what that outcome is going to be. So that's the first piece on selection and stratification. The second piece is in terms of outcomes. Now, I don't know if the day will ever come where the FDA will be accepting of a biomarker outcome in the sort, right? I don't think that we want to base it on functional MRI or anything like that, but that was the purpose of developing the tasks. The tasks have a direct relationship to these circuits. And if we're able to polish and clean up and train up the tasks so that they can do very, very well at identifying circuit level dysfunction, we can use them as a proxy. And I do think that down the line, the FDA will be much more receptive to having a cognitive assessment, a digital assessment that can be done in the doctor's office, on a tablet, online, what have you, be a reasonable outcome. The thing that you have to remember about the FDA regulatory process is that to this day, they still require a combination of not just cognitive. In the case of Alzheimer's disease, it can't just be cognitive, it also has to be functional, right? So you have to really change something about the way that the patient either thinks, feels, or is able to kind of act in everyday kinds of circumstance. So whether or not preventing memory loss on this task is going to reflect itself into, they're less forgetful when they go to the store, or they're able to balance their checkbook better, or any of the functional aspects of life, is still not clear. That's a really tricky part to get to. But given that a lot of the diseases that we're talking about don't have solutions right now, I do think that there's going to be an appetite to approve on the basis of cognitive and maybe not so much function. It's not there yet, but we're working on it. The FDA's got to kind of continue to develop its approach along with the science developing, right?
Yeah. Awesome. Thank you so much.
Of course. Nicole?
Hi, Mike. I just want to say thanks for an awesome talk as usual. I remember seeing some of this when I was a graduate student at UCI, and it's just amazing to see you take it and run with it, but not surprising. Anyway, my question is, you were talking kind of early in the talk about the goal of decreasing activation in CA3, is that right?
Yes.
So one of the things that we've been working on at UCLA is using functional connectivity guided neuromodulation.
Mm-hmm.
And I was wondering if you had, well, maybe it's a naive question. If there's like disintegration of the actual pathways, maybe brain stimulation is not going to be an effective intervention. But have you thought about it, or have you seen any work using brain stimulation, I guess broadly in terms of treating memory deficits, but more specifically in terms of improving pattern separation?
To be honest with you, I don't know the answer to that, Nicole. I mean, I think it's a really compelling question, and I've talked with Nanthi and others about why this is really important. If we can do something like perforant path stimulation, for example. So instead of trying to address the hyperactivity, can you override it by improving the quality of the input, the information coming in? Because at the end of the day, it's all about balance of information sources that are coming in. And the coding properties at the population level are going to reflect some balance or imbalance of those information sources. So if we can't control one, maybe we can just push the other one to be better, right? And I think that the data that so far I've seen out of UCLA is very promising that one can do that. But I can't put a mechanism to it. I can't say it's doing that by enhancing inhibition or improving, firing or any of that. We just know that there's a behavioral outcome. So I would love to see much more on the circuit basis. And what you're talking about with looking at functional connectivity, for example, and neuromodulation functional, that's exactly where we need to go. So I'm excited to hear that. I can't wait to talk with you more about what you guys are doing, because I think that's exactly the way of the future. And now there's so much more. I mean, it used to be that stimulation was like this thing that, wow, so invasive. I don't know if we really can go down that route. But now there's a lot of advances that suggest that you might even be able to do much less invasive types of stimulation and be able to get memory rescue. So I'm watching with excitement and anticipation. I think, Jesse, you've got a question here. Do you want me to read it, or do you want to ask it out loud?
Hey there. Sure, I can ask it out loud. So this is perhaps a bit much to ask, because this paper just came out today, and I don't know if you are aware of it or had seen a
No,
preprint.
I haven't seen this. I'm just reading.
Yeah, and unfortunately, because of the ongoing dispute between the UC system and Elsevier, I can't actually access the paper and haven't been able to find a PDF online. So I don't really know the story beyond the abstract. But anyway, Rodrigo Quiroga argues that this idea of pattern separation, which comes out of the computational models we discussed in fMRI data, he says that this is not supported by the single unit data, and argues for an alternative framework. But this will perhaps have to be a discussion for another time, because we need some chance to digest this new piece and see if it.
You've just made my day by putting this on my radar. So I needed something other than the election to think about. So I'm definitely going to try to get my hands on it, maybe email a couple of colleagues who still have access to Elsevier, see if they can send me a PDF. But I'm familiar with some of the theoretical work, and I've had conversation with Rodrigo before. But if he's got data, I'd love to see what he's talking about. So
okay, I'll leave it at that.
Appreciate it. Yeah.
So great. So actually, I had a list of questions, and I don't see any other questions from the audience. But I think in the interest of time, I could email them to you.
Anytime, sure.
I'll just ask one. So I like the shout out to David Marr, who has actually done obviously phenomenal work in cognitive neuroscience, but I know him through computer vision, and so I know these models have been now refined and now more advanced on, but yeah. So my question was probably a very naive question. I always had this question to know how to ask. The memory storage in hippocampus, is it more of a storage or is it more of a computation?
Yeah, so I mean, there's probably a couple different schools of thought, although I think everybody would agree that nothing gets stored in the hippocampus. I mean, maybe very little temporarily, just to be able to kind of store an online type of memory, but the real storage is happening elsewhere in the hippocampus, at least as far as indexing theory goes, right? Is that it maintains an index or a record of where the different parts of that memory might be stored throughout the cortex, but at least the eventual storage sites are always been thought to be the cortex rather than the hippocampus itself. And we know this to be true partially, at least, because if you take out the hippocampus, you still have access to a lot of the past memories. So presumably they have become largely independent of the hippocampus. I'm saying this knowing fully well it's a simplification, and there's lots and lots of different models that talk about this in different ways. We've hypothesized one of our own that suggests that there's trace interference, that the hippocampus is involved in the process of consolidation over time. So there may be a perpetual continuing role for the hippocampus with any sort of retrieval, even past remote memories. But that does not necessarily mean that it is really the storage site. It may hold on to a memory for a little bit of time, while, for example, there's an active attractor in CA3, but it's not really thought of as a storage.
Okay. So that means this pattern separation, for lack of a better word, I'm going to really simplify this, the algorithm for pattern separation that will happen in our brain. The parameters for that algorithm, will they be stored somewhere? So in other words, maybe the hippocampus is receiving inputs from all these different sites, and then it's eventually doing something.
Yeah, it's interesting. Shantanu, I think you're asking a very important question that not a lot of people can provide an answer to, but I will tell you, this is a shout out to one of my other students whose work I didn't talk about too much today. Sandra Gaddis is working in my lab in collaboration with Gary Lynch, doing some electrophysiological recordings in slice, in hippocampal slice, and trying to understand exactly what that algorithm is that allows for conversion of information from one subfield to the next. And one of the things that she's trying to do is figure out exactly what those parameters are. And how to, the computation is not always just, either separate or not separate. There's a threshold, and there's really kind of more of a sigmoid shaped function. And she's trying to identify all of that, but it is so difficult and so time consuming. And there are so many different aspects of that network that need to be controlled also, right? And this is still in slice. And the question is, okay, what about when you move to in vivo, where you have so many other influences? So it's the beginning of what I think is the sort of the algorithmic exploration that we're going on right now, but I don't have the best answers for you right now. Ask me again in a few months when she's ready to kind of, talk about it and put that model out there. But it's exactly the right question, is what are those parameters and where are they stored? And we suspect that some amount of that is stored in the hippocampus, because that just makes sense. I don't know that it would be getting it from the outside, but we have to demonstrate that.
Okay, great. So, and I had a bunch of lists of questions, and your talk was really inspiring. Actually made us think through a lot of things. So I'm going to email you those questions. So for now, and that's why I think we wanted to have you in person on the campus so we could just sit and talk and discuss ideas.
Would love that. Would love that. Let's hope the pandemic ends soon and we're able to get together again in person. I always love visiting UCLA, and you guys are more than welcome to come down and visit with us as well. But yeah, this is very, very engaging and lots of great questions. So
perfect. So let's thank Mike again virtually for his great talk.
Sure. Thank you all so much for attending, and it's great to see everybody, even though it's virtually, and hopefully we'll see each other soon in person.
And see you guys next time. This is the end. Bye.
Yassa, I have a question.
Oh, sure.
So I was wondering if, because ultra high resolution sounds like a pretty cool technique, but then exactly how high can it get? So can it resolve like individual cell layers from the internal cortex, like layer 2 versus layer 3, like spatially?
Yeah.
And then, okay.
Well, with the diffusion approach, I'll be very specific. It's 0.667 millimeters in plane, but they're thick slabs, because we're able to, we know that the perforant path structure doesn't really vary in that anterior posterior dimension. So we could afford to get thicker slabs, but then we have very high in-plane resolution. We've been able to do it with just under like sub-millimeter resolution also recently with some new and innovative high angular resolution tractography, and also look at it. But the question you're asking in terms of layers, I think could be better addressed not with diffusion, but really looking at this with very high resolution structural scans. And I do see that, on our 3T, for example, it's really difficult to get a resolution that is below, say, 0.5 millimeter. But some other folks have looked at this at 7T, and they're able to look at 200 micron resolution. And they're looking at some really nice separation across layers. Now, are you able to say layer 2 versus layer 3? Probably not. But like within the hippocampus, for example, you can pull out SRLM and say that's a layer that has different intensity values and is well organized, and we can pick up on that. Within the cortex, because 6-layered cortex, 4-layer cortex, pick your choice as you're going into the medial temporal lobes. Those very much look similar on structural scans. There may be a way to look at this with diffusion. We're probing that right now with these very high resolution zoom-in scans based on what we believe is dendritic orientation within those layers, but we're still not there yet. I mean, we're able to do it for the white matter, but looking at the gray matter and the entorhinal cortex, we can't do it. We can do it for hippocampal subfields, and that paper will be out soon. That's work from Steve Granger in my lab. But and I'm happy to send you some more work on that. He's able to pull apart subfields based on their diffusion properties.
That is interesting too, just because I didn't know exactly like how do you interpret FA within gray matter, and if cells are organized in a linear fashion.
Yeah, we've got some really interesting FAMD profiles that are highly differentiated across subfields, and only with this method. So if you look at it with just traditional DTI, it just all fuzzes together and you lose it. How to explain that is still tricky, and we're still grappling with that for his paper a little bit. How do you explain that a particular subfield has this profile of FA and MD versus another subfield? We're trying to use what we know about the anatomy, like the fact that there's granule cells in the dentate, so there's high densely packed kind of cell structure versus in CA3, but that's still without the postmortem histology on the same scans, right? So we don't have that direct one-to-one. We'd like to do that for sure, but it involves
Manual segmentation.
yeah.
Okay.
Yeah.
That's how you're getting your areas, is manual segmentation.
So yes, so we do have manual segmentations that we then are able to apply. We have now a deep learning algorithm that takes our manual segmentations in a training set and applies it to all the de novo scans, and that works really well. But for Steve's paper, I think he just used ASHES, which is works reasonably well and approximates subfield distributions reasonably well. We've been able to outperform that with some of the recent things that we've worked on, but like I said, ASHES is available and it works reasonably well. Far better than FreeSurf for subfields, I should say. FreeSurf for hippocampal subfields is not the most validated method, and it overweights the priors and makes it look really good. But the problem is when you try to validate it on the actual scans, it comes up with details that is not physically there.
Okay, I've seen that too, but the tractography results look better just because you have more surface area, or like it goes off of vertexes.
Yeah.
My follow-up question was then, have you looked at, if you can't look at individual layers within the perforant path that you do see, because you get this reduced or cortical thinning in ALEC, that may be predictive of memory impairment, do you also see less anterior perforant path versus posterior perforant path?
That's great. Okay, I'm going to tentatively say yes. And it's because I've seen some of the data on this in the lab already, and it's looking promising. It's looking like it's exactly that it's really the perforant path coming from the ALEC rather than the PMEC. But I don't have the full data set yet, so I can't commit to that. But that's exactly the right hypothesis to test. And like I said, our data so far seem to be in line with that, but I don't have the full data set to know for sure. So let's hold off on that question. I'll be able to give you a better question.
Yeah. Thank you very much.
Of course, of course.
So we will allow one more question from Emily.
Sure.
Before the,
okay. Yeah.
If you're fine, Michael. Yeah.
Hi. I'm wondering if the medial temporal lobe is thought to play any role in short-term memory or working memory. I think that's mostly the frontal lobe, but I'm just wondering whether any of your work is focused on that area and possible deficits with age.
Yeah, so let me speak to the first part of that, whether the medial temporal lobe plays a role in working memory. We believe that it does, and we haven't done the full thorough examination of this, but we're collaborating with Weiwei Zhang at UC Riverside. We actually have a grant that an R01 that he's the PI on to look exactly at that phenomenon. And there's at least one paper now that's in revision from this work showing clearly that not just the hippocampus, but even the dentate gyrus is involved in a working memory variant of this pattern separation task. Very, very exciting stuff. So I think we're coming to a better understanding that kind of defies the logic of the Squire et al. Model a little bit, that you get this beautiful dissociation of memory systems. Turns out memory is not so clean, right? And the brain doesn't care whether you call it working memory or short-term memory or long-term memory. If it's got any element of holding on to information the hippocampus wants in, it wants to be involved in some way. And we see this also in working memory. Whether that's compromised in aging, I can tell you that we have not done a thorough examination with that particular task in aging, but I could tell you that the differences that we see in our work are not driven by either perceptual or working memory versions, because when we do those controls, we see that older adults are not really struggling with it. Now, that said, those are not optimized to pick up on working memory pattern separation. The task developed way is. So that's why we want to apply those tasks to older adults before I can have a definitive answer.
Thank you.
Of course.
Great, fantastic. We have more questions after the talk ends. I love it.
Happy to answer any questions. Send them my way. All right, great to see you all. Thank you so much.
Thanks, Mike, again for coming virtually.
Of course.
Bye.
Of course. Take good care. Bye-bye.